Hello, my name is Tasia, and I'm excited to share with you the research I did this past summer on HPLC analysis of tRNA modifications in yeast cells. Our project centers on transfer RNA, also known as tRNA. As you may know, tRNA acts as the molecular bridge between mRNA and amino acids during translation, but what's often overlooked is how heavily modified tRNA molecules are. These chemical tweaks, like methylations and isomerizations, are essential for tRNA stability, folding, and decoding accuracy. When these modifications go awry, the consequences can be profound. In fact, defective or hypomodified tRNAs have been linked to disease states, including metastatic tumors. We focused on a yeast mutant cell strain lacking RRP6, which is a nuclear exosome protein involved in RNA quality control. In wild-type cells, RRP6 helps degrade faulty tRNAs, but in RRP6 delta mutants, that safeguard is missing. Our hypothesis was simple. If defective tRNAs escape degradation, we should see altered chromatographic profiles, specifically differences in modified nucleoside concentrations. To test this, we developed a reverse phase HPLC method using UV detection. We first isolated tRNA from yeast cells, digested it with nuclease P1, and dephosphorylated the resulting nucleotides. We then ran standards: cytidine, uridine, guanosine, adenosine, and pseudouridine on the reverse phase HPLC instrument here in the chemistry labs. Our mobile phase was ammonium phosphate and methanol with a gradient ramping from 2.5% to 40% methanol. The method proved sensitive and reproducible. Our calibration curves, seen in figures 2 and 3 respectively, at 260 and 280 nanometers showed strong linearity, and we achieved low limits of detection, down to 0.0039 milligrams per milliliter for pseudouridine, seen in Table 1 at the bottom of this poster, at 280 nanometers. In wild-type samples, seen in Figure 6, we observed clear peaks for all major nucleosides, including a distinct pseudouridine peak around 4 minutes. In contrast, the RRP6 samples, seen in Figures 7 and 8, showed lower overall nucleoside concentrations, and the pseudouridine signal was below quantifiable limits. These findings support our hypothesis. Without RRP6, hypomodified tRNAs may accumulate, but their degradation products are less detectable, possibly due to altered modification patterns or reduced stability. Our method not only separates and quantifies nucleosides effectively, but it also opens the door to deeper investigations into RNA modification dynamics in disease models. In closing, I'd like to thank the Virginia Ellis França Fund for Chemistry for supporting this work. I would also like to thank Dr. Klein for being my research advisor this summer, and Colin Levesque and Dr. Chatterjee with their help supplying the tRNA samples and overall biology help.